Hi everyone, I am Kaumeel Chokshi, a postdoctoral researcher at Tokyo Institute of Technology, Japan. My research area is microalgal biotechnology, and today I'll be discussing about bioprospecting of microalgae for sustainable bioresource development. Before starting my lecture, I would like to thank Dr. Nisikant Vase for inviting me on this Life Sciences discussion forum being organized by Botany for You. I have divided my lecture into 5 sections. First, I'll briefly mention my academic background. Then, I will discuss about introduction of microalgae, microalgal culturing in the laboratory, followed by applications of microalgal biomass. And in the last section, I will discuss my research that was conducted during my PhD. So first, let's move to the first section. So I had completed my MSc in 2010 from Sardar Patel University, Gujarat. And during my MSc, I had carried out dissertation at CSIR Niri Nagpur. As you know, CSIR is the Council of Scientific and Industrial Research, and it has around 38 to 39 laboratories in all over the India. After completing my MSc, I joined CSIR CSMCRI Bhavnagar, and in 2014, I was enrolled for PhD in the ACSIR, that is the Academy of CSIR. After completing my PhD in 2018, I received the fellowship from JSPS, and currently I am working at the Tokyo Institute of Technology, Japan. So let's move to the introduction. So whenever we go nearby water bodies, we generally see greenish layer floating on the top of the water. That is generally algae, and mostly you will see this kind of presence of algae in all the water bodies like lakes, ponds, rivers, ocean, rocks, etc. And it is believed that there are around 30,000 to 100,000 types of different algae. So this algae can be classified into two types. First one is the macroalgae and second is the microalgae. So macroalgae are very large and multicellular, as you can see in this picture, and it can be easily seen by the naked eyes, and it is known as seaweed. Whereas microalgae are very small, and it can be seen only under microscope, and it is also known as phytoplankton. So my research is related to microalgae. So microalgae are the oxygenic photosynthetic organisms, and they utilize light energy and nutrients and synthesize lipids, carbohydrate, proteins, pigments, etc. And all these products has the potential for use for different purposes like feed and food, nutraceutical, cosmetics, medicine, fertilizer, etc. And recently, microalgal biomass is considered as a renewable feedstock for biofuel production. There are several reasons for this. First one is the higher lipid and carbohydrate contents, which can be easily converted into biodiesel and bioethanol, respectively. Microalgae also has the faster growth rate and higher photosynthetic efficiency compared to the plants, and the demand of fresh water as well as land for their cultivation is comparatively lower than the plants. And microalgae also possesses a higher degree of environmental tolerance. So microalgae is considered as a valuable bioresource because it helps, there are several reasons. It helps in reducing the pollution. As I said, microalgae absorbs carbon dioxide from the environment and releases oxygen in the environment. The growth rate of microalgae is faster than the plants. Uh, if you if you want certain amount of biomass, the plants would take around months or sometimes more than 10 or 12 months. whereas the same quantity of biomass microalgae can produce within some days. So the growth rate of microalgae is faster than the plants. The requirement of nutrients is also very less, and microalgae can be cultivated in seawater as well as wastewater. So ideally, there is no investment of fresh water or nutrients compared to the plants. And in case of microalgae, there is a feasibility of large-scale cultivation. So if you want a certain amount of biomass, you can easily scale up using the open ponds or large-scale cultivation. So let's discuss about how microalgae is cultured in the laboratory. So when you are not dealing with the model organism and you want to isolate microalgae by yourself, so what you do is you go into the nature. So we can go and collect the microalgal samples and water samples of the water body and then bring those samples into the laboratory. In the lab, we can add those samples and provide some nutrients into the glass plates, and then we can allow it to grow. So this photo shows typical growth chamber for microalgae. It has lights from all four sides, and you can set desired temperature, whatever you want to wish. So after getting some growth, you can then isolate different species by conducting the isolation isolation using standard microbiological methods. Once you receive the isolated colony, you can then pick up and easily grow into the liquid medium. You can, for faster growth, you can also provide CO2, and then you can scale up from 1 ml to 10 ml and then 100 ml and then up to liters. So by this way, you can culture the microalgae into desired volume. So uh, once you have the highly grown microalgae culture, you can then separate the biomass and eliminate the water by several means. You can also use filtration, you can use centrifugation, and after these processes, you will get the biomass like this. So this biomass of microalgae has several applications. So let's discuss about where microalgal biomass can be used. So as I said earlier, the biomass contains lipids, carbohydrate, and proteins, and all of them has several applications. It can The lipids and carbohydrates can be converted into biodiesel and bioethanol. The high products from microalgae has the nutraceutical and pharmaceutical potential. Microalgal biomass can also be used as food and feed, and it can also be used for the fertilizers, cosmetic industries, and as well as to prepare the bioplastics. The high value products from microalgae such as beta carotene, pufa, lectins, phycocyanins, they have several usages like color for food and feed, vitamins, antioxidants, etc. And these are some of the examples where microalgae has been used in a commercial products like cookies, chocolates, energy bars, and then tablets, green tea, and your feed supplements, etc. So now let's discuss about what I had done during my PhD. So my PhD was focused on bioprospecting of green microalgae, Ectodesmus dimorphos. So Ectodesmus is the freshwater microalgae which was isolated from industrial wastewater polluted stream. And I had isolated this microalgae and we had characterized its morphology and identified as Ectodesmus dimorphos. So before moving on to my experimental part, I would like to briefly introduce some of the terminology which I have been using in the following slides. So I would like to focus on reactive oxygen species and oxidative stress. So any fluctuating environmental conditions trigger a series of physiological processes in the cells, and this generates reactive oxygen species. So as you can see here, imbalance in the electron transfer chain will lead to generation of superoxide radical in the cell. This superoxide radical can be converted into perhydroxyl radical, hydrogen peroxide, and hydroxyl radical. So this kind of ROS are, earlier it was believed that uh, they only damages to the cell and leads to the oxidative stress. But recently it was observed that various scavenging mechanism of the cells allows this ROS to be used as a signal which can control the growth and development of the algae. So cell has the cellular defense against the generation of this oxidative stress, and there are two types of scavenger: enzymatic antioxidants and non-enzymatic antioxidants. So enzymatic antioxidants contains SOD, catalase, APX, GPX, this kind of enzymes, which can eliminate these uh, reactive oxygen species into H2O2 and O2. And these non-enzymatic antioxidants like carotenoids, proline, and polyphenol, they can also be used by the cell to overcome the oxidative stress. So my aim was to understand the physiology and biochemical responses of the ectodesmos dimorphos under different abiotic stress conditions. So as you can see in this picture, there are various factors which affects the growth of the cell. This includes amount of the nutrients, availability of the nutrients, temperature of the environment, availability of light, light and dark cycle period, and light intensity, and then quality of the water, including wastewater, etc. So all these factors affects the growth of the cell. This in turn affects the accumulation of pigments, lipids, protein, and carbohydrate in the cell. And as I discussed earlier, any fluctuation in the optimum conditions in the cell leads to generation of this reactive oxygen species. So generally, there is a balance between reactive oxygen species production and reactive oxygen species scavenging. But once this balance is impaired, the condition leads to oxidative stress, and then it can also kill the cell. So for my research, I have focused on temperature, salinity, as well as nitrogen, and growing the cell in the wastewater. So today, for this lecture, I would discuss about my research on temperature stress as well as wastewater. So this slide shows the response of Ectodesmus dimorphos to cultivation temperature. So as we all know that temperature is very crucial factor for the growth and physiology of microalgae. And for any organism, it, it can lead to cell death if the temperature is very, very high compared to their adapted temperature. And temperature directly influences the photosynthesis process and the biochemical contents of the cell. So for my first major experiment, we had chosen to grow the cells at different temperature. Generally, in the laboratory, we grow the cells at 25 degrees centigrade. But for my study, we had chosen 25, 35, 38, and 40 degrees centigrade for 15 days of growth period of this culture. And simultaneously, we had also performed two-stage cultivation in which microalgae are grown at 25 degrees centigrade for certain time. And then for remaining time, they are transferred to 35 and 38 degrees centigrade. For both these experiments, I had monitored the growth parameters and various oxidative stress biomarkers in the cell. So as you can see here, the slide shows the pigment composition of the cell. So the green, we should focus on the green part. The accumulation of chlorophyll A and chlorophyll B was the highest at 35 degrees centigrade, and the carotenoids were also highest at 35 degrees centigrade. This suggests that compared to 25 and 38 degrees centigrade, 35 degrees centigrade is the suitable temperature for this strain. Also, the ratio of chlorophyll A by B as well as carotenoids by chlorophyll A by A plus B is lowest at 35 degrees centigrade. So these ratios are used as an indicator of the stress condition. So the higher is the ratio, the higher is the stress for the cell. So at 35 degrees centigrade, as you can see, this ratio were the lowest. Similarly, when the cells were exposed from 25 to 35 for the 9 days of stress, you can see that the the chlorophyll content was highest. So this suggests that the better the time, the better is the adaptation of the cell at 35 degrees centigrade. Coming on to the biomass production and biochemical composition, the biomass production was also highest. As you can see here, it was the highest at 35 degrees centigrade compared to this 25 and 38 degrees centigrade. And also the lipid and carbohydrate contents were almost same as that of the 25 degrees centigrade. Coming on to the response of the stress biomarkers, so the stress biomarkers indicates the level of the oxidative stress in the cell. So the higher the value, the higher is the stress condition for the cell. So in this case, as you can see that at 35 degrees centigrade, the level of S2O2 in the cell was the lowest. The activity of the antioxidant enzyme catalase and APX was also the lowest at 35 degrees centigrade. So this suggests that the level of oxidative stress is minimum at 35 degrees centigrade. So all these findings led us to propose that at 35 degrees centigrade, Ectodesmus dimorphos can easily adapt and well acclimatize compared to the 25 and 38 degrees centigrade. So if you want detailed results for my study, you can refer my paper that is published in Bioresource Technology. So once we found that the optimum temperature for the cell is 35 degrees centigrade. So 35 is comparatively higher than 25. So this led us to check that thermotolerance ability of the cell. So in case of microalgae, the thermotolerance is defined as the ability of the cells to tolerate the higher temperature, that is generally around 10 to 15 degree more than their optimum temperature for up to 24 hours. So as in our case, 35 degrees centigrade was the optimum temperature for the cell. We decided to choose 40 and 50 degrees centigrade for up to 24 hours. And there are only very limited microalgae that has been reported to have this kind of ability. So we design an experiment in which cells were cultivated up to their mid log phase and late log phases at 35 degrees centigrade. And later on, they were hit stress at 45 and 50 degrees centigrade for 8 and 24 hours. And here we monitored the changes in the pigment composition, biomass, lipid, and carbohydrate contents. So as you can see here, the chlorophyll content before hit stress. So before transferring the cell was 36.17, and after transferring at 45 degrees centigrade, the chlorophyll content was increasing. Whereas at 50 degrees centigrade, the content of 36 was decreased to 22 and 21 after 8 and 24 hours. So this suggests that cells has the ability to adapt at 45 degrees centigrade for up to 24 hours, and that is why we found increase in the chlorophyll. Similarly, carotenoids were also increased from 4 to 6 at 45 degrees centigrade, but at 50 degrees centigrade, the carotenoids were reduced to such a level that they were not detected. So for both mid-log and late-log phases, we found that at 45 degrees centigrade, the chlorophyll contents increases, whereas at 50 degrees centigrade, the chlorophyll contents decreases. So next we extracted the lipid from the cell, and then we fractionated the lipids. So the lipid has three classes: neutral lipid, glycolipid, and phospholipids. And when we analyze the lipid class of 45 degrees centigrade and 50 degrees centigrade, so we observed that at 45 degrees centigrade, the column A suggests mid-log phases cells treated at 45 degrees centigrade for 8 hours. So when the stress of 8 hours was extended to 24 hours, the neutral lipid were increased from 64 to 78%, and this had led to decrease in the glycolipid contents of 23% to 8%. Whereas at 50 degrees centigrade, as you can see here, the changes in the neutral lipid content was not so significant. It was around 65 and 67. Similarly, for late log phase cells, the neutral lipid content after 8 hours was 62%, which had increased to 73% after 24 hours. And at 50 degrees centigrade, rather it has decreased. So this suggests that cells can remodel their membrane lipids, and this is one of the strategy for the survival of the cell. So these findings led us to propose that Ectodesmus dimorphos is a thermotolerant microalgae, and these findings were published in the Journal of Applied Phycology. So as we had found that this strain is very interesting in giving the response at 45 degrees centigrade, we next designed an experiment in which we had uh, we had uh, we had again exposed the cells at 45 and 50 degrees centigrade, but at this time we had monitored the level of oxidative damage through ROS, as well as the response of enzymatic and non-enzymatic antioxidants at different time interval, like 0, 4, 8, 12, and 24 hours, to know the exact strategy and the difference in the strategy of the cells at high temperature between 45 degrees centigrade and 50 degrees centigrade. So I'm not discussing in detail, but what we had observed is represented in this one figure. So at 45 degrees centigrade, we had observed that the chlorophyll and carotenoids content were maintained or rather increased sometimes, whereas the proline and polyphenol contents were increased. Whereas at 50 degrees centigrade, the chlorophylls were decreased and the proline and polyphenols were also decreased. And as I discussed previously, we had observed remodeling of the lipid. So this might lead like adaptation of the cells by remodeling of the membrane lipids. So we concluded that at 45 degrees centigrade, the defense response of the cells is active. That is what giving him the ability to resist the heat, and maybe that is why it is showing the thermotolerance. Whereas at 50 degrees centigrade, these decrease in the proline and polyphenols, as well as decrease in the pigments, as well as there is no remodeling of the membrane lipid. So the cellular defense response gets impaired, and this has led to the toxicity of the cell. So these findings were very well correlated uh, with the live and dead ratio of the cells. So as you can see here, for a purpose of example, I have shown the accumulation of proline and polyphenols in the cell. So as you can see at 45 degrees centigrade, the proline was higher than 0 hours, that at 24 hours, whereas at 50 degrees centigrade, the proline content decreased and it led to like minimum or maybe not at all. Again, for the late log phase cells, the proline content increased, whereas at 50 degrees centigrade, the proline content decreased with the time. Similarly, the polyphenol content was also increased with the time, whereas at 50 degrees centigrade, it decreased significantly. So this kind of observation was one of the reasons that we thought that this algae is showing the thermotolerance at 50 degrees centigrade and not at at 45 degrees centigrade and not at 50 degrees centigrade. So uh, for for detailed reference, please refer my recently published paper in Physiologica Planetarium. So once we found that this microalgae has the ability to tolerate high temperature, we have characterized its response for temperature as well as nitrogen starvation as well as salinity stress. But here, due to time constraint, I am not discussing all my results. So now let me move to the application part. So we have utilized this microalgae for the phycoremediation of dairy wastewater. So you might be knowing the phycoremediation is the use of algae for the removal of the pollutants from the water. So in this case, we had used the dairy industry wastewater, in which water is a key processing medium for the various processes, including cleaning, sanitization, heating, cooling, and floor washing. So before this study, the all other studies which were reported for the phycoremediation of various wastewater, so all those studies were focused with the additional steps, like the wastewater was treated for the pH adjustment, or it was diluted, or it was mixed with some other kind of wastewater to minimize the to minimize the pH effects, etc. But here in our study, we collected the raw dairy wastewater, and without any process, directly I used it as a medium for the cultivation of Ecotodesmus dimorphos. So for this purpose, we collected the dairy wastewater from the local dairy, Sarvottam Dairy. It is located in Siho, Bhavnagar. And this dairy has the very huge turnover of the milk, and it mainly focuses on the packaged milk, buttermilk, and ghee with the brand name of Amul. So what we had done is we had just filtered to remove the suspended particles, and initially we had stored before carrying out the experiment, we had stored this dairy wastewater at 4 degrees centigrade. And when we performed the experiment, we directly used that filtered dairy wastewater, and then we performed the experiment for 8 days at 35 degrees centigrade. So what we had observed is on day 0, the COD level was very high, like 10 times in the required limit for the release of this dairy wastewater. So it was like 2600, and the ammonical nitrogen level was also very high. So when we grown this Ecotodesmus dimorphos, we observed that within 4 to 6 days, it can consume most of the nutrients from the cell, from the water, and it can reduce the level of COD from up to 90 times. And the ammonical nitrogen was also reduced from 277 to almost zero at 6 days after the starting of the experiment. So this data shows the growth of the cells. The highest biomass was observed at after 4 days of cultivation, and it was corresponded with the cell counts of the microalgae. It was also highest at after 4 days, and the biomass which was harvested after 8 days, it contains uh, the lipid and carbohydrate content was comparable than the control. And the after 8 days of cultivation, the dry cell weight of this dairy wastewater grown biomass was 5 times higher than the normal DG11 culture, which is used as a reference. And this biomass contains around 25% lipid and around 30% of the carbohydrate. So we represented our theoretical conversion yields, like the annual dairy wastewater which would be generated by this industry is around 64 million liter. And if we consider the total yield of 840 milligram per liter, so we could generate like 53 tons of the biomass from this one dairy industry. So this 53 ton contains around 25% lipid. So this can be converted into biodiesel, and the lipid extracted biomass, which was further utilized for its value addition, but it contains around 29% of the sugar. So this sugar can be fermented to ethanol. So all in all, we can convert this lipid and carbohydrate into biodiesel and bioethanol respectively. So theoretically, we can convert, this can lead to 273 gram of biofuel per kg of the biomass. And additionally, we can also reduce the CO2 from the atmosphere. So as I mentioned, we had used this residual lipid extracted biomass for its value addition. So what we had done is uh, we had we had prepared the water extract of the biomass, and then this water extract was used for the synthesis of silver nanoparticles. And after synthesizing the nanoparticles, we characterize is using TAM, SAM, and elemental mapping. And we observed that the particles has the size of around 2 to 15 nanometer. So altogether, this demonstrate an integrated approach of phyco nanotechnology. So first, we collected dairy industry wastewater, and then we grown the microalgae in that water. And after utilizing it for biofuel potential, we then the remaining biomass after the extraction of lipid, we then utilize for the synthesis of silver nanoparticles. And those silver nanoparticles have the scavenging ability, which was demonstrated by the DPPH and ABTS free radical scavengers activity assay. So if we integrate such kind of approach, we can develop a sustainable biorefinery from microalgae. So both of these studies are uh, published in Bioresource Technology and RSA Advances individually. So this was all about my research during my PhD. So after finishing my PhD, I received the ACSR based PhD thesis award. So this award was uh, given to 3 students out of 204 students who successfully defended the PhD program of ACSR. And as you can see, my father is receiving this award from Dr. Harshvardhan at CSR headquarters. As I was in Japan, I could not attend this program. So at last, I would like to acknowledge my mentors, Dr. Arup Ghosh and Dr. Sandhya Mishra, for their guidance and valuable suggestions during my PhD, and financial support from CSIR in the form of CSIR Senior Research Fellowship, and all my friends and family for their support throughout my study. So that's it for my presentation today. So if you have any question or if you want to contact me, you can leave a comment below, or maybe you can get the email ID from the organization and you can contact me. Thank you very much for listening to my lecture. All the best.